So that's just to wake everybody up.
Yeah. So this is basically John November's fault, but so.
Blame you, John.
Yeah, so this was mostly to wake you all up after lunch, but it's actually super relevant for what I'm going to talk about today. So hopefully that took you back a couple of years to when those Harlem Shake videos were all over the internet. And what this was showing is two different MCMC algorithms. The first one is Metropolis Hastings, which just like moves in a random direction, and it's basically looking at does the density get higher, and if it does, then I go in that direction. The second thing, the Harlem Shake part, is an algorithm called Hamiltonian Monte Carlo, which uses the gradient of the density locally. And as you could see, HMC is much better at exploring these difficult densities that represented those letters. And a bunch of what I'm going to be talking about here is how using gradients when you're trying to do many different things, so optimization and sampling and also variational inference, really helps out a lot in optimization. If you don't do it, you're going to have a bad time. Okay, and this is going to be sort of a mix of sort of tutorial flavor and then also illustrating things with examples from my own research. So I want to start out with saying a little bit about sort of my view of how I like to think about statistics. And this has a bit of a Bayesian flavor, but if you're uncomfortable with that, you don't need to think about that part too much. So I like to think about whatever data you observe from your experiment as having been generated by some stochastic process. So you can call that a model, if you like. And so all this equation is saying is my data D was sampled from some stochastic process Q. And then statistics is all about, given some observed data set, what can we say about the stochastic process that generated the data? If you're being Bayesian, then this means you're trying to think about what's the posterior distribution over Q conditioned on data. And Bayes' rule would tell you that you can break this down as thinking about what was the likelihood of the specific data set given some instantiation of the stochastic process, and you might also have a prior on the stochastic process. So usually we're thinking about relatively simple sort of parametric models when we do this, but I just want to point out that that's not always the case, and there are lots of fun examples where there are more interesting generative processes that people use. So this is an example from CVPR, which is one of the big computer vision conferences a couple of years ago, where this group from MIT, the generative process they're actually using is sampling from a distribution over three-dimensional shapes. In this case, it's a Gaussian process that gives you the profile of this shape, and then it's lathed in 3D and given some spatial position and orientation, and then that 3D object is passed through a renderer to give you a 2D image, and that would be the data that you observe. So this is a much more sort of complicated stochastic process than we normally think about, but modern Bayesian inference are actually methods where you can take that sort of image data and do inference to try and ask questions like what was the shape that generated this data. So one class of stochastic processes that are convenient and sort of tractable to work with are probabilistic graphical models. So these are, the graphical part here does not mean that there are pictures involved. It means that the structure of the model is specified by a graph. And in these graphs, nodes correspond to latent variables or parameters, and edges tell you about the dependencies between those latent variables. I'm going to just talk about directed graphs here, which you can also get called Bayesian networks. So for some given node in the graph, what the graph structure tells you is that under the generative process, the distribution of x here is dependent only on its parents in the graph. So that's what this is saying. And then we could write down the whole joint probability distribution of the whole graph by this factorization over all of the nodes. I'm going to assume fixed structure throughout this talk because learning structure is really hard. There's a great literature on trying to do it. But if you can avoid it, it's definitely nice. There's also a big literature on trying to do probabilistic inference in these sort of models. So things like belief propagation. If you know what the Baum-Welsh algorithm is for doing the forward-backwards path of Baum-Walsh for an HMM, you sort of already know what belief propagation is. So belief propagation is just the generalization of that forward-backwards path to trees. There's a further generalization to loopy belief propagation. So this is if you have graphs that are not acyclic, that have cycles. For here, I'm going to, so usually when people talk about Bayes nets, they often get talking about discrete latent variables. Everything I'm going to talk about today is going to be using continuous latent variables and parameters. And the reason for this is we get to leverage gradients. So remember back to our Harlem Shake example, gradients are really useful for effectively exploring a parameter space. So I'm going to use as a use case for these ideas work we've done recently on looking at splicing and alternative splicing regulation. So to remind you about splicing, human genes in higher organisms like humans are made up of exons and introns. So the exons are the parts of the gene that are actually code for protein. So transcription gets you a precursor messenger RNA, and then splicing is the process by which the intronic regions are removed to give you a mature messenger RNA, and then of course those are translated to proteins. Alternative splicing is what we call it when there are sometimes alternative choices in what gets removed as intronic regions. So here I'm showing a second isoform for this gene where exon 2 wasn't included, and of course that's going to result in a different protein. To give you a motivating example for this, I want to introduce you to the tau protein. So tau is a protein that helps stabilize these structures called microtubules. Microtubules are really important in neurons, particularly in axons. So axons you can think of as the output channels of neurons, whereas the dendrites here are the input channels. And axons are pretty long typically compared to the cell body. So an axon might be like 1,000 microns, whereas a cell body would typically be on the order of up to 100 microns. Since axons are so long, it's challenging for cells to move nutrients and proteins up and down that long distance. And the solution to this is these microtubules, which you can think of as the railway network of the axons. And then these proteins called kinesins can carry vesicles holding the nutrients that are needed up and down the axon. Tau consists of 14 exons, and this is not that unusual for a human gene. The typical human gene has around 9. And there's various of these exons that are alternately spliced. I'm going to focus on exon 10, though, because this is particularly important, as you'll see. So isoform 3R excludes exon 10. Isoform 4R includes it. And the reason these are called 3 and 4R is that the 3R version ends up with 3 microtubule binding domains, whereas the 4R version ends up with 4. As you might imagine, if you have more microtubule binding domains, you end up with higher microtubule binding affinity. It turns out that in healthy development, in the fetal brain, you see almost exclusive expression of the 3R isoform, whereas in healthy adult brain, you see balanced expression of these 2 isoforms. So this is a situation where there's a functionally important difference in healthy tissues during development. What's also interesting is that in various neurodegenerative diseases, including in particular frontotemporal dementia with Parkinsonism, you see an imbalance of these 2 isoforms. So in particular, here you actually see an increase of the 4R expression, but there are also diseases where this goes the other way, including Alzheimer's. Okay, so that example suggests that splicing is important both in healthy development and also in disease, so how can we quantify it? And the way I'm going to be talking about doing that is using RNA sequencing data. So work we've done recently has involved better approaches for quantifying splicing variation using an algorithm called leafcutter. And then I'm also going to talk about more recent work trying to understand the genetic basis of splicing dysregulation. So existing methods for quantifying splicing variation from our short-read RNA-seq data basically fall into 2 categories. The first are isoform ratio estimation. The idea of these methods is that you start with some set of known isoforms for a gene, and then you try and look at how the ambiguity in how reads are assigned to those isoforms, and use statistical estimation to try and sort of bootstrap up, typically using EM, an estimate of the level of each of these isoforms. There's really great work in this area, but whatever you do, it's a really statistically challenging problem, and as a result, the estimates you get tend to be pretty noisy and not robust to small changes in the input data. Another approach that people use a lot is to, yeah.
What happens when the isoforms are not known?
Yeah, so that's definitely a problem. So there is work that tries to like add to the known set of isoforms as like a pre-processing step, and then do this using that extended set of isoforms. So you can sort of try to address that. But even if you know the full set of isoforms, especially for larger genes, there's going to be uncertainty between the levels. So the second main group of methods try to calculate exonic percent spliced in. So the idea here is that you would focus on some particular exon. So typically you would think of this as being what's known as a cassette exon, where it may or may not be included in the isoform. And then think of these exons to the side as being constitutive. So that means they're always included in the isoform. Then you can look at reads that unambiguously support the inclusion or exclusion of that exon. So that would be these reads that map to the exon itself, support its inclusion. These spliced reads that map across the two exons also support its inclusion. And then these three spliced reads at the bottom would unambiguously support that it was not included. And then I'm not going to go into the math, but you can account for read length and exon length to try and use these numbers to get an estimate of this. The problem with this is that while it works well for exon skipping events, there are lots of more complicated splicing events. For example, 3' and 5' extensions, multiple exons being skipped at once, mutually exclusive exons, and complex splicing events representing a combination of these things. And in all of these settings, the assumptions that you use to calculate the exonic percent spliced in don't really work anymore. So our idea was to think of a third way of doing splicing quantification. And this is to quantify the level of intron use instead of exon use. It sounds like a really simple change, and in many ways it is, but it comes with various conceptual and computational advantages. So the biggest really is that introns are uniquely defined by splice reads. If you observe a splice read, you know exactly what was cut out of the precursor messenger RNA. So that directly defines the unit that you're going to quantify. Unlike exons, where if you see a read in an exon, you don't know what the boundaries of the exon were. By using these spliced reads, it's also very easy to quantify how much each intron was excised. Just count how many splice reads correspond to that intron. Makes you much less reliant on existing gene models because the data itself is telling you what you're going to quantify. And it also lets us model all these more complex splicing events, which exon skipping quantification did not. There's still a question of how do we group introns into the units that we're going to consider for analysis and normalize. So we can't just measure intron excision counts themselves and use those directly because that would be completely confounded by total gene expression. The way that we do this is to define splicing components, and I want to show this graphically because I think that's the easiest thing. So imagine this gene where we have these three constitutive exons, but then there's a cassette exon, an alternatively spliced exon here, and a 5' extension on this final exon. The spliced reads for some region might look like this. And the splicing components that we define correspond to building a graph where the nodes in the graph are the splice sites, so the starts and ends of the introns. And the edges in the graph are going to correspond to observed spliced reads, so observed junctions. So the components that you would get in this situation would be these two, sitting on either side of this constitutive exon. And then the quantification we're going to think about is how much are each of these intron used relative to the other introns in the same component. And each of these more complex splicing events that I showed you would end up corresponding to an individual splicing component. And I'm just showing here the spliced reads that would let us quantify what's going on in each of these situations.
So is it the case that it's always just the constitutive exons that define the boundary of the components? That was that example, right? Just make sure.
Yes. So there are. Yes, I think that's probably the best way to think about it. Yeah.
Basic question: how do you identify the spliced reads?
So we don't deal with alignment. We let star, or we've used the Lego a bunch, like some good RNA-seq aligner. We don't deal with that side of it. And obviously we let those. So those, is maybe worth saying on that point, we do let the aligner look at GenCode transcript models, right? Because you do a lot better if you do that. But all of the good methods, right, will also do de novo splice junction detection. So having defined these splicing components and found and counted up our splice reads supporting each junction or intron, we'd like to be able to compare between conditions. For example, you'll see if you look at heart and brain samples that this RBFOX1, which is an important splice factor itself actually, you see strong differential splicing where in brain, this exon is used at the expense of this exon, and the other way around in heart. So to test for these sorts of differences using our counts, we're going to use a directionally multinomial generalized linear model. The reason we do this is the multinomial is going to represent a likelihood that's looking at choices of which introns were excised. The Dirichlet lets us model over dispersion, and the linear model part means that we can include whatever confounders we want and also test for things like, does the covariate representing which tissue I'm in improve the likelihood of the data? So to explain this model, I first need to tell you what the Dirichlet distribution is. So the Dirichlet distribution is a distribution over distributions. So I'd like to give you a minute to try and wrap your head around that. So if you don't think about it as a distribution on distributions, you can think about it as a distribution on probability vectors. So pi here is a probability vector. So all the elements are greater than 0, and they sum to 1. In other words, they live on the k-simplex. The notation here I'm using is just, so subscript dot just means summation over the elements. And it turns out the expectation of this distribution is just given by normalizing this parameter vector. So that also implies that an alternative parameterization that you can use that's a bit more convenient is to set A here equal to Cp, where C is a scalar called the concentration parameter, and P is a probability vector that's just the expectation of the distribution. This is showing what a Dirichlet distribution looks like on the 3-simplex, so if you have probability vectors of length 3. So if A is the 1 vector, then that corresponds to the mean being at a third, a third, a third, and the concentration parameter being 3. And as you increase the concentration parameter, you see that the mass of the distribution is concentrating around the mean, hence why it's called a concentration parameter. And of course, you can also move the center of the distribution. OK, so the reason the Dirichlet distribution is nice to use is it's the conjugate prior for the multinomial distribution. So if you make a little hierarchy where you sample a probability vector pi from the Dirichlet distribution and then sample counts y from a multinomial with probability vector equal to pi, then integrating over pi, you get that the y's are directionally multinomial distributed. And the y's are going to correspond to the counts that we model for the spliced reads. And the functional form is pretty simple, which is also nice. OK. If you take c to infinity, that concentration parameter, then the directionally multinomial just reverts to being a multinomial. OK, so how would we actually use this in our linear model? So the counts yi, I is going to index over individuals or samples, and j is indexing which intron we're looking at. And we're also going to need the total number of counts across all the introns in a cluster, in a component, and these covariates x. So all we're going to do is say that our counts y are directionally multinomial distributed, and do the normal regression, x in a product with a regression coefficient beta. But we stick these through a softmax function just so that these p's are going to be a valid probability vector. The graphical model for this is just shown here. So what I'm showing shaded gray are observed variables. So x and y are both observed. We want to learn beta, and we also want to learn C. I'm abusing graphical model notation a little bit here, because P is really a deterministic function of these two. But it's sort of helpful to see the structure in the model this way. OK, so we have a model. And so now I can talk to you about how we would use, yeah.
So one of the things about the directional multinomial is the covariances between your introns now are going to be negative.
Yes, that's true.
Do you think that?
Well, it's negative in that I mean, all it means, right, is if you're a bit more likely to use this intron, then it means I'm a bit less likely to use this other intron.
But then you can't consider them independently the way that, like, biologically may happen, because biologically you can consider an intron to be spliced independently of the next intron, possibly.
Possibly. I mean, honestly, the covariant structure in any of these could be quite complicated, right? Because we're not doing, I mean, there are methods that try to do the things where you actually look at, like, what was the path that was taken through the splice graph. And it's just really hard.
But you're considering each component separately, right? So if you have
Yeah.
one intron, you pretty much don't have the other intron. So I think you actually want that, don't you?
I think it's hard to say from the biology what's like the right thing to do. But from a testing point of view, it's possible we lose a little bit of power from not modeling that covariance, but I think it would be conservative if anything. OK, so probabilistic programming. So the idea of probabilistic programming is that you take your probabilistic graphical model, for example, the one I just showed you on the previous slide, and we're going to write it down as what's called a probabilistic program. So this is going to be some sort of domain-specific programming language that lets us write down that model in a way a computer can understand. Once you've done that, there are various tools now that will let you take that program and internally generate the computation graph. And once you have the computation graph, so sorry, I should say this is the computation graph to calculate the likelihood or log joint, if you're thinking in a more Bayesian way. Once you have the computation graph, you can use what's called automatic differentiation to work out how to calculate gradients automatically. And once you have gradients, you can, for example, do maximum likelihood using optimization, say using LBFGS is a popular and good performant optimization algorithm when you have gradients. So this would be an example of doing point estimation. But as we saw with the Harlem Shake example, you can also use gradients in Markov chain Monte Carlo methods, particularly through Hamiltonian Monte Carlo or its variants. The example I'm going to show you here is using a probabilistic programming language called Stan, which is out of Columbia. So this is Bob Carpenter and Andrew Gelman are kind of the lead PIs on this, I guess. It's extremely well developed and maintained. There are interfaces for R and MATLAB and Python. And the really awesome thing about this is you write your model out in a very sort of clean, high-level syntax. And once Stan goes and does these steps for you, it's writing C++ code for you. So you never have to write any C++ code, but you're going to get super efficient C++ code under the hood that's going to do all the optimization for you. What does one of these programs look like? So this is a slight simplification of the directionally multinomial GLM. So you need to tell Stan about what inputs it's going to get. So it needs to know what's the sample size going to be, what's the number of covariates, how many introns are there, what's the actual input data going to be, so covariates and counts. We also put a prior on the concentration parameter, so we need to know what that prior is going to be. We need to know what the parameters in the model are going to be. And then actually setting up the model, all we have to say is that the concentration parameter is going to have, we put a gamma prior on the concentration. And then for each sample, the counts are directionally multinomial distributed with the form of the model that I gave you earlier. And once you have this, what Stan under the hood will be doing is taking that probabilistic model represented as the probabilistic program, calculating its computation graph, which sort of looks quite similar to the graphical model, right? Except that the thing it's trying to calculate in the end is the likelihood, L. And what it's then going to do is work out how to use automatic differentiation to automatically get gradients. So automatic differentiation, which is also known as backprop for neural nets, involves a forward pass through the computation graph. So this is just starting, say, here with the x, beta, and c, and propagating forward through the graph to the likelihood. And that means you're going to calculate all the intermediate variables along the way. And then a reverse pass where you start at the likelihood and work back to each parameter. And the rules for that is actually pretty simple. It's just going to use chain rule. So for any, starting out at L, you'd propagate backwards through the graph. So for any variable x, what you need to do is just the multivariate chain rule. So the gradient of L with respect to x is just a sum over the children of x, which I've called y here. And hopefully you all remember chain rule. So the partial derivative of L with respect to the particular child that we're looking at, and then we also need to know the partial derivative of the child with respect to x. Makes sense? How am I doing for time, Angel? Where are we going?
That's calculated numerically.
Yes, right, yeah, exactly. So as you're going back, every time you're numerically evaluating this, numerically evaluating each of these, and then adding them up. Yeah. Yeah. So if, in a way, I thought about it, in a way it would be clearer to show this if you put, the bar to say that you're actually evaluating this at the current parameters, but I thought it looked messy, so yeah, right.
Is it doing numerically like with a little perturbed epsilon, or is
No, it's all exact, yeah.
Exact.
No, it's all exact, yeah, which is part of what's nice about it, is you don't have to worry about what those epsilons would be. How am I doing for time? 15 minutes? Okay, I'll do like a brief version of this. So I wanted to show sort of what this would look like for the particular model I'm showing, which is here. I've broken this down a little bit more just to say that the probability vector here is just a softmax of this G, and this would be multiplication. So here, G would be X transpose beta, and then P is just the softmax function of G. So the way in the algorithm that it would propagate gradients backwards here would be to do, so for, say, we're going to focus on trying to get the gradient with respect to beta. So we'd first get the gradient with respect to P, this. And that's going to be you're going to have to go and get the derivative with respect to P of the log directionally multinomial likelihood, right? So this is going to have like this. I'm not going to work that out because there's a lot of gamma functions. And then as you propagate back, so to get the derivative with respect to G, that would just be, there's only the one child of G, right? So this would be the derivative of P with respect to G, and then the derivative of L with respect to P. And this thing is just the first derivative of the softmax, which you can do as a homework assignment. And then then we propagate back one more. So we want the derivative of L with respect to beta. So that's just going to be the derivative of L with respect to G times the derivative of G with respect to beta. And this thing is just X, right? Because from here. So it's pretty simple. We just start calculating this numerically and then keep propagating back through the graph. Does that make sense? Yeah, that's a question.
So in that model specification you gave, does the gradient have to be calculated across all the parameters, or can you do like an alternating?
You could in principle. Usually when pretty much everything I've seen done in this sort of framework just does everything at once. I guess it's simpler. I mean, in general, I think if you don't have a reason to do something like coordinate descent, it's probably more efficient to do this, right? Because say you're using LBFGS, right? LBFGS is going to, as it runs along, it's sort of learning this approximation of the Hessian, which means you're going in a more efficient direction than if you did coordinate descent. So I think you're better off doing everything together when you can. I guess there might be times when there are computational savings from that. Okay. For leafcutter itself, we use LBFGS and use likelihood ratio tests to compare whether a specific covariate should be included in the model or not. One thing we've used this for is looking at differential splicing between tissues in GTEx. And we find thousands of differentially spliced introns between tissues. And if you focus on the most differentially spliced introns, then you see a pretty clear clustering of samples by tissue type, which you don't see if you just do all introns. And we also find that if you look for those, which introns are differentially spliced in human across different mammalian species, then for a significant proportion, you actually see conservation of those differences across species in orthologs, suggesting that for a fair proportion, there are functional roles. We've also used leafcutter quantifications in transcriptome-wide association studies, which I think Haki will be talking about tomorrow. But I wanted to give you the basic idea here. So a TWAS is, the idea of a TWAS is, consider the situation where you have a GWAS cohort where you have some trait, but you don't have any molecular phenotypes, but you do have genotyping. So you have cis SNPs around each gene. And you also have some reference cohort, say GTEx, where you have, say, RNA sequencing data. So the idea of a TWAS is to learn a predictive model of gene expression going from genotypes, local genotypes, to gene expression, and then to use that predictive model to impute expression in the GWAS cohort. And then you can think about associating those predicted expression traits with your high-level trait. You can also do this using just GWAS summary statistics, which is convenient because that's usually what you have access to. And while people usually just consider total expression, what about also thinking about splicing? So the example I want to show you for this is using Parkinson's disease, and this is work that my collaborator Yang Li has been doing recently. So this is using data from the RNA-seq data from the Common Mind Consortium, and large Parkinson's GWAS. And what Yang found is 18 and 26 genes respectively associated via expression and splicing. And I really wanted to show you this because the gene that comes up the most strongly is MAPT, which is microtubule-associated protein tau, so the example I showed you at the beginning. So this work, the TWAS results suggest that the dysregulation you can see in disease in terms of splicing can have a strong genetic component. So we wanted to try and think about how to predict the effects of genotype on splicing. The way we've been doing this is to think about trying to predict for a given 5' splice site which 3' splice site will be used. So here I'm showing an example of a 5' splice site in this gene where there are various exons that are spliced to, and also some sort of noisier splicing events here. And the model we're going to use is a deep convolutional neural network, but this is a fully convolutional network, which means that if you like, a sequence-to-sequence model. So the input is DNA, or if you want the precursor messenger RNA sequence, which is passed through convolutional layers, but there's no max pooling here, which means that the resolution isn't, the genomic resolution isn't decreasing, although of course you are losing some base pairs at the end. And once we have, after some number of layers, we have a vector of outputs, and those are again going to get passed through a softmax, and we're going to use directional multinomial likelihood again to model where splicing events occur. So it turns out that actually what happens when you fit neural networks is very similar to the picture I showed you earlier of using a probabilistic program. So I actually, for this work, I used Theano and a library that sits on top of Theano called Lasagna. The popular thing to use these days is TensorFlow. But the idea is really very similar in that what languages like Theano and TensorFlow let you do is write down your neural net as what I would think of as a probabilistic program, and then go through these steps of getting a computation graph, a gradient, and then still doing maximum likelihood, although in the neural network setting, of course, typically you're working with the scale of data where you want to do stochastic optimization, so things that only look at mini batches of data at a time. So typically stochastic gradient descent or something like ADAM. So here's the lasagna code for this model is called Grasshopper because it's meant to be like a grasshopper like hopping along the precursor RNA. So again, it comes out pretty simple. You need to define the parameters that are going to be in your, or the variables that are going to be in your model. So the X here will end up being a one-hot encoding of sequence. The Y's will be the RNA-seq counts. And the core of the model, right, is all here, just these convolutional layers. We do this batch normalization, which helps with not overfitting and with convergence. The directional multinomial is still easy to write out. And lasagna will let you do stochastic gradient descent updates all on the GPU. Yeah. I've been doing fixed lengths for this. In principle, you could do not, but just for practical. I have done both actually, but for the most part, I've done. Yeah, I should say that for this, I've been doing 30 kb, like from the 5 prime.
What are the samples?
The samples here, yeah, that's a good point. So the idea with this is that we're trying to generalize across the genome. So you're learning. So effectively, a sample is really for us is every 5 prime splice site is like a sample. And then there are multiple individuals, but they're effectively almost just giving you more read depth of like where are the splicing events rather than really being the n that you would care about.
And would it matter if you did this for like train this for every splice site or splice site within a region?
Yeah, I think it would. I mean, there's a lot of the models that are doing best here end up having a receptor field of a sort of 800 base pairs, and having sort of like 6 or 7 layers. So there's a lot of parameters. So you want to be generalizing across as much genomic context as possible. Yeah. Okay. So yeah, I'm not going to. And the computation graph that Fiano would be making in this situation looks like this. So here, this is the hidden layer in the neural network before getting the softmax to go to the likelihood of the loss. And as you can see, this looks pretty similar to what we saw previously, right, for the directionally multinomial GLM. I'm not going to talk too much about performance, but I can show you something on that if there's time later. But you can do fun things with this, like in silico mutagenesis, to look at what sequence features seem to be driving predictions. And we pull out a bunch of features that are sort of known splicing regulatory elements, things like branch points, the fact that exons tend to be GC-rich, and also that to be a good 3' splice site, you need a downstream 5' splice site that the spliceosome can use. We've also started using this to look at predicting the effect of rare or de novo variants on splicing outcomes. We've been testing this out using GTEx, since they have whole genome sequencing for most of the samples. This, for example, is a rare variant that changes this G to a T just downstream of this AG dinucleotide, and results in actually like greatly increased use of this as a 3' splice site. Okay, so I just wanted to wrap up by saying that I think this framework for thinking about fitting statistical models is quite appealing in its generality. I've mentioned Stan and Fiano. There are a bunch of probabilistic programming languages. Winbugs was one of the earliest ones. Church tries to be extremely general in terms of fitting Bayesian non-parametric models as well as parametric models. Infer.net is a project I worked on out of Microsoft Research in Cambridge. Edward is maybe the most cutting edge thing right now. This is out of David Bly's lab, and they're doing. Edward I really should have put on both of these because Edward can do both probabilistic graphical models and neural nets and combinations of these two. And actually Edward works on top of TensorFlow. PyTorch, people seem to really love it. I haven't used it. You can even think about modeling, so structural equation models, which we don't see too much, I feel like, in more computational genomics, but so these are used extensively in social sciences, for particularly for asking questions about causality, sometimes in a well-founded way. And they have their own software, actually. The one I'm aware of the most is called OpenMX. But again, it works on the same principle of taking if you write down a probabilistic program and then go through these steps. And I just wanted to wrap up by saying, if you don't want to do Markov chain Monte Carlo, because you don't know what to do with all the samples you get anyway, and you don't want to do maximum likelihood because you want to be a bit more Bayesian than that, there's also stochastic variational inference methods. And these are actually built into both Stan and Edward. And what these do is let you try and fit an approximate posterior distribution to the true posterior. So the object you get at the end would be something like, if your data was all continuous, if your parameters were all continuous, for example, would be something like a multivariate Gaussian distribution that's trying to fit to be as close to the true posterior as possible. OK, that's everything. Good timing.